Enzymes, a protein with catalysis properties due to its power of specific activation used to speed up chemical reactions in the body, also called biological catalysts. Reusable, one enzyme can be used many times. Specific, the enzyme has a specific folded shape so that the active site only fits its intended substrate. Anabolic reaction, reactions enzymes bring the substrate molecules together ex photosynthesis and protein synthesis. Catabolic reactions, the enzyme active site affects the bonds in substrate so they are easier to break. Food digestion. Lock and key hypothesis assumes the active site of an enzyme is rigid in its shape. Only molecules of that shape can fit. Induced fit hypothesis suggests the active site is catalytic and only assumes its catalytic conformation after the substrate molecules bind to the site. The active site molds itself into the shape of the substrate molecule. Tertiary structure, active site has a specific shape due to of proteins. Active site, a change in shape of the proteins affects shape of the blank and the function of the enzyme. Denatured, enzymes may become blank in harsh environments such as incorrect pH and harsh temperatures. Environmental conditions, cofactors and coenzymes. Enzyme inhibitors, what affects enzyme activity? Temperature, increasing the blank make molecules move faster. Biological systems, very sensitive to temperature changes. Increase the rate of reactions. Temperatures, lowering the activation energy. Enzymes can blank without increasing the blank by a new reaction pathway. Enzymes create a shortcut. 108 to 1011. Non-enzymic, enzyme-controlled reactions proceed blank times faster than corresponding blank reactions. Extreme pH levels will produce denaturation. The structure of the enzyme is changed, the active site is distorted, and the substrate molecules will no longer fit in it. The effects of telephone. Ionization, thus change in blank will affect the binding of the substrate with the active site. The temperature coefficient, Q10 equals the increase in reaction rate with 10 Celsius rise in temperature. Doubles or triples, for chemical reaction the Q10 equals 2 to 3. 30 C. For most enzymes the optimum temperature. C. Most enzymes fully denatured at inorganic substances, blank, and vitamins are sometimes needed for proper enzymatic activity. Iron, pick up oxygen, blank must be present in the quaternary structure E equals hemoglobin in order for it blank. Completive inhibitors are chemicals that resemble an enzyme's normal substrate and complete with it for the active site. Non-competitive inhibitors, inhibitors that do not enter the active site but bind to another part of the enzyme causing the enzyme to change its shape, which in turn alters the active site. substrates, the surface on which an organism grows or is attached. Products, different molecules that were converted through substrate. Catalyst, substance that speeds up the rate of a chemical reaction. Metabolic pathways, series of chemical reactions catalyzed by enzymes. Products, enzymes convert the substrates into other distinct molecules. Activation energy, the minimum quantity of energy which the reacting species must process in order to undergo a specified reaction. By reducing the energy needed for reactants to come together and react, how do enzymes lower the activation energy of a reaction? Shapirians provides an environment that is best suited for folding of the proteins. Secondary structural level, all proteins have at least a blank. Primary structure, black lines between the amino acids represent peptide bonds between carbon and nitrogen. The R group, tertiary structure depends on blank that each amino acid has. Sequence of amino acids and the hydrogen bonds it is able to form, the difference between the two secondary structure of proteins.